my name is Kaihim Wong. The topic of my research is moving toward faster life measurements of axon diameter. So if brain is a computer, axon are the wires that intertwine that each other allow information to exchange from different parts of the system. So by looking at the title, we might ask, why would we want to measure axon size? Do axon size vary from person to person like their height? Are they unique as fingerprints? Do they vary across disease or disorders? Trust me, I have those questions too at the beginning. So while the knowledge and experience that we accumulate from the past about biology, anatomy, and physiology can, for example, facilitate the chiropractic system to help patients who suffer from physical pain. And the knowledge also helps pharmaceutical scientists to develop specific pills to heal sickness, but it's not enough. Complex neurological disorders such as schizophrenia and autism are still unsolved puzzles. In no doubt, studies from off-field are focusing more on this one giant topic. A few studies combined with autopsy results suggest abnormalities of axonal integrity can be an early sign of developing schizophrenia. So by establishing a way to measure the width of the axon, allow us to conduct more studies on neurological disorders. So based on the size and dimension, we're not yet able to measure the properties of axon consistently. Even with advanced magnetic resonance imaging technique, there's not one that is clinical except at the moment. So from them, we have chosen oscillating gradient-based sequence to develop into a clinical usable technique, mainly for two reasons. It does not need high gradient strength to reach a short diffusion regime in a typical human axon dimension, and also minimize the eddy current effect on a human body. But like the other developing methods out there, we're still suffering from a long scan time, and that prevents us from having live mice scanning session to make cross verifications with mice axon electron microscopy. So the second column of my poster is mainly focusing where the signal is coming from and how they get analyzed. Because of the abundance of the hydrogen nuclei in water and fat inside our bodies, MRI is able to provide an extraordinary full-body soft tissue contrast, especially in the brain. But we cannot determine the axon width by looking at the image because of the resolution limit. Also, the conventional MRI gives no contrast for the water content in and out of the axon. So this is why you see the term diffusion appearing in the poster. Instead, we're measuring the signal from moving water molecule, which we call diffusion. So diffusion time can be categorized into long and short based on gradient frequency by considering axons as non-permeable cylinders, and the pictures in the middle, like on the top of the second column, shows a cross-section of the geometry with three types of water compartments: free, hinder, and restricted. So we have the model down below to structure the last two compartments, and the free water is neglected. But those compartments are also interchangeable if we're in different diffusion time regime. To have a better sense of what I've just said, imagine diffusion MRI is taking snapshots of the water movement. In an ideal case, if I decide to take a snapshot that is just short enough to let the only restricted water to hit the wall obstacle, there would be no difference between hinder and free. So to optimize scan time for imaging, we need to look at two main parameters. So first one is gradient amplitude, which is suggested by Morgan and taken care by Melissa up on the offer list. And the second one would be the gradient frequency, which is my focus on this project. With less frequency used or sample, I decide to use only the restricted model for signal interpretation. And again, this traces back to the example that I just gave. It's only theoretically valid if I have an optimal range of frequency to take the right snapshots. So by relating Einstein diffusion equation with oscillating gradient-based sequence parameters, we can possibly reduce our imaging by a factor of 12.5. But plot on the right shows an intermediate correlations between expected and inverse value with consistent error bars. On the downside, we must have a prior knowledge of the size of the axon to do our experiment though, which in our case is to know which frequencies to use. If this method is further improved and shown to provide consistent results without losing much signal to noise ratio, we can possibly peel one more layer out about the relationship between schizophrenia and axonal width, With that being said, this gives the research groups in off-field an extra tool to conduct experiments from a different perspective. At the end, I would like to acknowledge this project is funded by NSERC, and I would like to say thank you to all my colleagues in the lab, as well as Dr. Martin for guiding and helping me along the way. And I'm happy to take any questions.
Dr. Bai, do you have any questions?
No, I'm good on this one. Yeah, thank you.
All right. Dr. Gorbet?
Thank you. So I was just trying to understand a little bit where you're going to take that a little bit further. So now you've been able to kind of develop this model that allows you to reduce the scanning time in mice. Is that correct?
Correct.
And so I guess I have two questions in a way. So yeah. Then, you know, how would you, you know, schizophrenia is, as far as I know, and I don't know a lot about some of those diseases, it's more of a human disease than maybe a mice disease. So how would you intend to kind of use that to study in the mice? And second, I guess for the human, it'd be great if you can actually reduce the scanning time. So is that also, do you think that's applicable to helping with those studies that are done on humans?
Correct. That's a great question. So actually, for our main purposes of this project is to actually quantify, like see whether we can quantify the axon width in anything so that we can move on to human. But then schizophrenia is just an example, but the study shows there's a relationship between axon integrity, for example, like demyelination, axonal width decrease, or any kind of thing as abnormal in the brain can cause schizophrenia. But that's another story though. But we would like to go from the perspective of measuring axonal width, therefore provide an extra tool for the scientists to go on from a different perspective to look at the disorder or any kind. But I hope I answered that question.
Yeah, no, that's great. Great. Thank you.
Thank you.
Dr. Gutierrez?
Yeah, I was just wondering the reason behind why your study is going into like a more faster live measurement of the axon diameter. I think I heard you mention that the importance of this axon diameter in terms of...
Right, axon diameter, right? Yeah, sorry. Sorry, can you repeat the question?
So what's the reason why you need to have like a more faster live measurement?
What? Sorry, you mean
why do I... about moving
why do I... Oh, yes. Yep. Okay, so yeah, I have to give the background story about what's the imaging time before doing this experiment. So before, it takes 3 days to scan a sample because we have to do 20 samples in frequency and 6 sample in gradient amplitude, which is 1, 0, and the other are the gradient contrast. So that in total is 120 sample, and we have to do it 3 times to reduce the signal to noise ratio to put into the model. But then if I want to scan a mice or a living object, 3 days is not relevant. So we want to cut that down, time down to 15 minutes or less, just so there's no movement artifact or actually discomfort of the patient, and the reliable of the data as well.
I see. Okay.
Yeah. Sorry, I didn't give the back of my story.
Thank you.
Thank you, Kaihim.
Thank you.
I actually have one more question.
Sure.
Is that okay? So,
yep.
So just to, because I'm starting to understand a little bit more. So then, because that's where potentially, that's where the validation I haven't seen, and maybe I missed that. But have you been able then to look at that your model was able to reduce the time, and if you had, mice that had a smaller axon, you were able to detect that compared to one, well, like, within the brain? Or have you done some validation yet, or is that the next step?
Yeah, due to the COVID thing that I wasn't able to conduct any validation experiments in the lab, but I do have data back in the days that where someone else acquired in the lab, where I have simulation data, I have celery simulations data, garlic stem as well. Yes. So
Okay,
yeah, in answering your question, no, I haven't do any live measurements, but the model is theoretically valid with the sample I sampled like we sampled before.
Okay, thanks.
Thank you.